Namaskar, Swagatam. We would like to welcome you all on our 520th milestone. I am really overwhelmed with the kind presence of so senior dignitaries. आदरणीय अग्निहोत्री जी प्रणाम, आदरणीय के सिजोरिया सर प्रणाम, आदरणीय कामिनी धीमान जी प्रणाम, आदरणीय महेश शर्मा जी, प्रमोद वार्गी जी, नमस्कार। नमस्कार मैडम, स्वागतम। रहिएगा, आज छोटी छोटी सी करने लगेंगे वेबिनार। तो मोहता जी प्रणाम, देवजीत और ठाकर जी आज आसाम से हमारे साथ जुड़े हुए हैं। आज क्वालिटी कंट्रोल ऑफ आयुष पाउडर एक बड़ा ही महत्वपूर्ण सब्जेक्ट है, जिसमें हमारे शॉर्ट नोटिस के उपरांत भी डॉक्टर सविता जी और डॉक्टर गुरु प्रसाद दोनों ने हमारा निमंत्रण स्वीकार किया। We would like to communicate our sincere thanks to both the speakers and senior dignitaries in the audience. Without wasting much time, I would like to request Professor V.K. Agnihotri sir for his initial blessings.
Thank you, sir. Thank you, Professor Pawan Sharma ji. It's a great pleasure that we are stepping in for 520th time just to celebrate our International Research Societies, Ethics Societies webinar, and that too with a very vital topic, that is the quality control of the powders, the churn of our pathy. So it's a great pleasure that we are having our two distinguished guest speakers. They will throw light over the possibilities and requirement, necessity regarding the quality control over our Ayurvedic preparations, especially in the powder form. So I welcome our both the guest speakers, Dr. Madam Sataji and our second guest speaker. Though it's very cold here in North India, so we would like to retire early as much as possible. So our idea is this, that at the time of the welcoming our guest speakers, our approach will be that how they manage to maintain the quality of the preparations, because we all know that it is the rainy season where the viratva or the efficacy of the powders loses. So what may be the expiry date, or what may be the life of their preparations. So with these words, I welcome our guest speakers and also our very senior persons like Mahesh Sharma ji and our Guwahati person, along with our KK Sijoria sir. It's our pleasure that they are sparing minutes for us. So thank you very much. And I welcome all of the guest speakers along with our senior dignitaries, those who have joined our today's webinar. With these words, I back this presentation ceremony to Professor Pawan Sharma ji, so that the time will be saved and we will be enlightened. Thank you, sir.
मैं आज विष्णु प्रसाद शर्मा जी नहीं है, मैं आदरणीय कामिनी धीमान जी को दो शब्द कहने के लिए, फिर उनके दर्शन नहीं होंगे आखिरी में। आदरणीय कामिनी जी, स्वागतम।
नमस्कार सबको, बहुत बहुत नमस्कार। अग्निहोत्री जी बहुत बहुत
जी नमस्कार।
आपको सुनने में तो बहुत मतलब
मैडम मैं आपका एक वाक्य अगर रोक दूं यहाँ पर, आप विश्वास नहीं करेंगे मेरे लिए ये कितनी गर्व की बात है। अभी 1-1.5 महीने पहले एक दिन आप जुड़ी थी और किसी ने, हमारे ही गेस्ट स्पीकर्स ने ये कह दिया, अरे धीमान मैडम को तो आप बुला ही नहीं रहे, धीमान मैडम को बोलने मौका दीजिए। उस दिन बाय हार्ट में कह रहा हूँ, मैं ये जान पाया कि आप मैडम धीमान जो है हमारे के धीमान साहब की वाइफ है, जो हमारे बीएचयू मिस्टर शाला के प्रोफेसर है, और and he happens to be my very senior student there in 1985 when I was assistant professor in Paparola College. He when he came to know that I am there in BHU, I was taking the PhD viva, he rushed to meet me, and rest of the faculty members, they were considering him a very senior person and professor and all that. And when he met me wholeheartedly, it was a great surprise to me that he is here after CCRS. And on that very day, I came to know that you both are a couple. Wonderful. My special affinity with you in that way. So thank you so much, madam. Please continue.
हाँ, मैं तो आपको इसी वेबिनार से ही जुड़ी हूँ, पवन जी के सौजन्य से ही
तो
मैं यहाँ पे जुड़ पाई हूँ।
आपको ये कहानी नहीं मालूम ना? आपको नहीं पता ना?
जी नहीं
आप इतना प्यार करती, इतनी रिस्पेक्टफुली मिलती बात करती थी, और मैं भी वो तो खैर हमेशा रहेगा ही, लेकिन हमारे केएस साहब के जुड़ने से तो मैं मजा ही आ गया। प्लीज कंटिन्यू।
हाँ जी, और जितने भी उपस्थित सारे सीनियर डिग्निटरीज हैं यहाँ पर, उन सबका मतलब पवन जी ने तो स्वागत कर ही दिया है। मुझे भी अच्छा लगता है, कभी कभी मैं जुड़ती हूँ, जैसे मैं हमेशा बोलती हूँ, आई एम इरेगुलरली रेगुलर। पवन जी ने मुझे तुरंत ही बोल दिया कि आप जुड़िए रहिएगा, क्योंकि मैं जुड़ती हूँ फिर लास्ट तक हमेशा नहीं रह पाती हूँ।
लेकिन in gynae this word is not expected, irregularly regular.
But I am the irregularly regular audience of this series of webinars. And इतने अच्छे स्पीकर्स पवन जी लेकर आते हैं, इतने अच्छे टॉपिक्स वो हमेशा लेकर आते हैं। और जब मुझे मौका मिलता है कि चलो ठीक है, अभी मौका है, पवन जी का मैसेज है, अभी थोड़ी देर पहले मैसेज है, मैंने तुरंत फटाफट ज्वाइन कर लिया। मैं नहीं, उनका मैसेज है, जुड़ते हैं, देखते हैं, बहुत अच्छे स्पीकर्स लेके आते हैं पवन जी, और बहुत अच्छा लगता है हमें सुनना। तो आज का जो टॉपिक है मेरे लिए, तो इट इज रियली न्यू टॉपिक। जो भी आप अभी आज आपने बोला कि ये टॉपिक है, मैंने कभी इसके बारे में नहीं सुना, तो इट इज प्रिविलेज टू लिसन योर स्पीकर्स। थैंक यू पवन जी।
थैंक यू मैडम, थैंक यू वेरी मच, थैंक यू वेरी मच। नाउ आई वुड लाइक टू इनवाइट Dr. Savita Bhonsle, in charge of the pharmacy of KLE Belgaum. Welcome, welcome. Dr. Savita ji.
Thank you, sir. Myself, Dr. Savita Bhonsle, working as a pharmacy in charge in KLE Ayurveda Pharmacy, situated in Karnataka, Belgaum. And I have a 14 years experience in the pharmacy production supervisor cum pharmacy in charge. Sir, today as my topic is quality analysis of powder. Sir, is it audible?
Yeah.
Sir, actually churna means we'll think maybe it is a single drug or a combination of the drugs. That is a multiple combination herbal mineral drugs that we can consider it as a churna. In Ayurveda, churna is a fine powder of a drugs and applied to the powders prepared by a single drug or a combination of more drugs, which is prepared by mixing clean, finely powdered and sieved drugs. The term churna cannot be considered scientifically valid if the drug tested had not been authenticated and characterized in order to ensure reproducibility in the manufacturing of the product. Moreover, many dangerous and lethal side effects have recently been reported, including direct toxic effects, allergic reactions, effects from the contaminants and interactions with herbal drugs. Nowadays, that guduchi we got so many issues with the guduchi, that liver, this one, but they didn't mention which species of the guduchi they have used for the nowadays that corona time. We found that liver toxic of the guduchi and all. This is because of the species. In order to assess the quality, purity, safety, and efficacy of the drugs, it is essential that standardization of any herbal formulations should be done based on the analysis of their active properties. Testing of Ayurvedic preparations using scientific methodology adds to the quality and accuracy of the product. Now, what quality controls we should follow with the churnas on the basis of the pharmacognostic, physicochemical, pharmaceutical, microbiological, toxicological, spectroscopical, and chromatographic parameters to confirm the identity, quality, and the purity. Here, so many drugs are there. There are testings for the analysis of the powder, but it is not compulsory to do each and everything for each drugs. But standardization need for standardization, at least basic quality analysis they should do. Standardization of the Ayurvedic formulations is an important step for the establishment of a consistent biological activity, a consistent chemical profile, or simply a quality assurance program for production and manufacturing of herbal drugs. WHO guidelines for the assessment of the safety, efficacy, and quality of the herbal medicines are a prerequisite for the global harmonization. Then we'll start with the churna. Earlier what the people, acharyas, they used to do, they will go themselves, they select the genuine raw materials. So there is no need of the standardization and all because there is no cultivation, there is no adulteration or any substitution. But here nowadays, as I am in pharmacy, I found there is more adulteration, more foreign materials along with the raw material. It will come. So here the question arises the quality analysis. First, we'll start with the determination of the foreign metals and organoleptic evaluations. Foreign metals, when we used to get, when we ordered the raw materials for the dealers, for to increase the weight, they are adding some substitute or some, they are making some adulteration like a sand, stones and all. So we should determine that foreign materials. For example, we should take 100 grams of the sample was spreaded in a thin layer and using a lens, magnifying lens, we should separate that foreign materials and we should calculate that percentage of the foreign materials, Now, second comes the organoleptic characters. These are all the physical parameters like color, odor, taste, appearance, and texture of the ingredients and formulation samples evaluated manually using the sensory organs of the body, standardized using a reported methods. Here, for example, color. Sometime of the for example, the sample of the Manjistra, which is Maharashtra, this one, it is having a light pink color compared to that of the Nepali color of the Manjistra. When we'll boil that Manjistra, Maharashtra, this one, we'll get light color compared to that of the Nepali. Here the color, but we should follow some standardizations and even odor, taste, odor. For example, the shunti, some it will affect the oil. Sorry. Then physical parameters. The physical parameters are bulk density, tapered density, Hassan's ratio, Cairns index, angle of the repose. By evaluating the physical characteristics of the churnas, the bulk density means it is the ratio of the given mass of the powder and its bulk volume. It is determined by transferring an accurately weighed amount of the powder sample to the graduated cylinder with the aid of the funnel, and it is calculated by bulk density is equal to mass of the powder divided by untapped value in a grams or the ml. Second one is the tapped density. It is measured by transferring a known quantity of the powder. Example, 30 grams, we are taking that powder into a graduated cylinder and tapping it for a specific number of times, and the initial value was noted, and it is calculated by mass of the powder. Tap density is equal to mass of the powder divided by tapped volume. The Hassan's ratio, it is indicated the flow of the powder, the flow property of the powder, and the ratio of the tapped density to the bulk density of the powder is called as a Hassan's ratio, and it is calculated by tap density divided by the bulk density. And compressibility index, it is the propensity of the powder to be compressed based on the apparent bulk density and tapped density. The percentage compressibility of the powder can be determined using the formula like tapped density minus bulk density divided by tapped density into 100. The angle of the repose. These are the basic formulation, basic analysis we should do for the physical analysis of the powder. In angle of the repose, the internal angle between the surface of the pile of the powder and the horizontal surface is known as the angle of the repose, and it is calculated by tan minus 1, height of the pile divided by the radius of the pile. Now next comes the physico-chemical parameters. These are all basic parameters we should follow for the analysis of the powders, like ash value, extractive values, loss on drying, pH values, swelling index, particle size, acid insoluble, water soluble, extractive values in percentage, that in alcohol soluble, water soluble, like this. The determination of the total ash can done as 2 grams of the sample was accurately weighed in a tared silica dish and kept in a muffle furnace at the temperature not exceed 600 degrees centigrade until it was free from the carbon. And it is calculated by weight of the total ash divided by air dry drag into 100. And determination of the acid insoluble ash. The total ash obtained was boiled for 5 minutes with 25 ml of dilute HCl. The insoluble matter obtained was collected on an ashless filter paper, washed with hot water until the filter rate is neutral and ignited to a constant weight. And it is calculated by weight of the acid insoluble ash divided by air dry drugs into 100. The water soluble ash is the ash obtained in the determination of the total ash was boiled for 5 minutes with 25 ml of water. The insoluble matter was collected on an ashless as that of the acid insoluble ash we should make, and it is calculated by weight of the water soluble ash divided by air dry drug into 100. The same method we used to do with the extractive values. The same for the determination of the water soluble extract, that is water soluble calculated it as a water soluble residue divided by air dried drug into 100. That is in percentage we should calculate it is. And determination of the alcohol soluble extract as that I already explained, and it is calculated by alcohol soluble residue divided by air dry drug into 100. And determination of loss on drying, 2.5 gram of the sample was weighed and placed in a tared evaporated successive weighing corresponds to not more than 0.25%. The determination of the pH is the powder sample was weighed to about 5 grams and immersed in 100 ml of water in a beaker. The beaker was closed with the aluminum foil and left behind for 24 hours in a room temperature. Later, the supernatant solution was decanted and into another beaker, and the pH of the formulations was determined using a calibrated pH meter. The determination of the particle size. For this, 25 grams of the sample was placed in a sieve of suitable nominal measure. Here, commonly we are using 80 number of the sieve, and sieve was shaken for not less than 30 minutes in horizontal direction and vertically by tapping on a hard surface. Weights of the amount remaining on the sieve were taken and determined the particle size. And it is calculated as weight of the sample taken tared into a weight of the sample remaining on the sieve and divided by the weight of the sample taken into 100. The powder microscopy. Why it needs the powder microscopy? The churnus was taken and was examined macroscopically to identify the various features like phloem fibers, parenchyma, starch grains, endosperm, calcium oxalate crystals, oil glands, etc. by using various reagents. The diagnostic characters such as the presence of the pollen grains, tracheids with spiral thickening, simple fiber, and oil glands indicate the presence of lignified trichomes, rosette and prismatic crystals, lignified fibers, and epicarps indicate the presence of goksura. The goksura, the future, the powder, it contains all these materials, and this can be done by a microscopy. Stone cells, mesocarps, parenchymal cells, vascular fibers, and trichomes indicate the presence of arm-like. These are some of the examples I have taken. Corn cells, the presence of the cork cells, stone cells, trichomes, and scleriform xylem vessels were suggestive of the Guduchi. These are the examples what we can see under the microscopes. And next comes to the phytochemical analysis. Phytochemical analysis, that presence of the alkaloids, presence of the saponins, presence of the cardiac glycosides, presence of the anthraquinone glycosides, carbohydrates, phenolic compounds, tannins, etc. These are the phytochemical screenings we should do for the powders. These are the basic. If we want some further analysis, then we should go for the thin layer chromatography, HPLC, LCMS. These are for, as per the WHO GMP, we should follow these some of the standardizations. And here for each standardization, we are following some APA book. But some standardization, they are not, we have used huge number of the tuners, and some are not, which are not mentioned in the API. then as we are in pharmacy, we should make ourselves in internal this one, we should make some standardizations and we'll keep this for the comparative. In thin layer chromatography is particularly valuable for the qualitative determination. TLC is a technique in which the solute undergoes distribution between two phases, a stationary phase acting through absorptions and distribution between the two phases, a stationary phase acting through the absorption, and a mobile phase in the form of liquid. The absorbent is a relatively thin uniform layer of dry finely powdered material applied to a glass plate are the most commonly used. Separation may also be achieved on the basis of partitions or a combination of partitions, and absorption depending on the particular types of supports in preparation, and it uses with the different solvent systems. TLC is an important tool by which the quality control and fingerprint of the herbs can be maintained. It also helps to identify the presence or absence of the individual herbs in combination herbal formulas. Means in combination, if they have not added some other, if they have missed, as a pharmacy is commercialized, they may miss some powders that can be identified through the TLC. I will give here, for example, in Shunti, there is a presence of gingerol, which we can identify with the TLC procedures. Here, the sample the powder sample extracted with the chloroform, and the extract was concentrated with the dissolved in a chloroform, and that is runs through the TLC. And even the piperine, the presence of the piperine can be identified by the RF-AYUSH 0.40 using a TLC procedures. Even the moisture content, for example, the shatavari, the guduchi, it is easily hygroscopic, means it absorbs the water according as per the seasons, that the moisture content can be calculated. Along with this, we can WHO GMP, even they will find the test for the heavy and toxic metals. There are some of the parameters which does the analysis of the lead, cardamom, mercury, arsenic. That is as per the limits, as per the ASU pharmacopoeia. There is some limits for all these heavy metals, and if you pass that heavy metals, then the that powder will get rejected. That can be calculated by using some analysis. For test for the metals or non-metals like a magnesium, carbonates, aluminum, iron, and chloride, limits as per the ASU pharmacopoeia. Now, nowadays the question arises about the pesticide residue study. This is the new one, because nowadays cultivated raw materials we are using for the powder, which contain because of the commercial because of the money matter, they are using some pesticides to get it grow or to make it a more So there is also some limits for the using the pesticides. So again, when the raw material will come, if we will get any doubt with that, then we should make a analysis for the pesticides like a organochlorine pesticides, organophosphorus pesticides, and pyrethroids pesticides. And there is also limits as per the ASU pharmacopoeia. Then the further advanced technology we found to do the analysis of the quality control for the powder, that is a Fourier transforms infrared spectrophotometric, that is FTIR. The FTIR is profiling spectra of all three batches of the means for standardization we used to make for the we should select the same 3 tuners, and we should make it for the standardization in the range of 4000 to 400 centimeter using a JASCO FTIR. Nowadays they are using the more this JASCO FTIR spectrophotometer, and here also some standards they are given how to do and all. The again more advanced technology if you want to do, then you should do for the high performance, that is HPTLC, high performance thin layer chromatography analysis, and LCMS, that is a liquid chromatography mass spectroscopy. For more investigations we can use all this one, and thermogravimetric analysis. The thermogravimetric analysis is used to determine the total weight change in the churna formulations during the thermal treatment. An accurately weighed quantity of the sample was heated in a aluminum crucibles with a heating rate of 10 degrees per minute from the room temperature to 1200 degrees centigrade in a nitrogen atmosphere using a thermogravimetric analyzer. The biocurve yield was calculated by mass balance. The data were interpreted using a software, that is a STAR-A software. Now, finally, the microbial contamination of the microbial contamination we can find, because as some of the raw material, some of the tunas, they are hygroscopic, and as they are hygroscopic, they are also get the easily the microbial contamination. So the API has given some limitations for the microbial load. For example, total bacterial count, they have given some limitations, total yeast and mold, and basic microbial load we are finding like the E. coli, streptococci, or streptococcal ARES, and pneumonia agumentis. These are some of the basic bacterial load we should check with the which comes under the qualitative analysis. Finally, the shelf life. As in Ayurveda earlier, the authors told that 6 months is the shelf life for the tunas, but as now Government of India has made it into a 2 years. But some of the tunas which practically I am seeing that Shatavari and all, that easily they will get contamination, or easily they will get spoiled by becoming it a hardened, or some growth will found on the some of the tunas. So shelf life we can't depend upon the 2 years. Even for example, triple R, if we'll keep for the longer period, means more than the 1 year, I found that it will come a very thick and some of the white patches grown on the that tunas. So shelf life, I think, depends upon the tunas which we have selecting, which we have preparing, and we internally we should make some standardization for that tunas. For example, triple R this month only we can use. For Shatavari this much month we can use. Some of they are not contain much that we keep for the longer periods. With this, I will close my presentation.
Thank you, Dr. Savita. Your voice is not coming, Savita ji. Excellent communication. Agnantri sir.
Sir, thank you so much, Dr. Savita. Mostly, really you touched all the sectors which are very essential to be known for all the persons, those who are attached with the manufacturing of the Ayurvedic medicines, especially the powder form. Rightly, you told that first of all, the impurities should be detected with the help of the, say, microscopic study, and all the impurities, especially impurities, they should be taken out. And for that, so many ways and means you told that it should be made very clear. And then comes it's a matter of self-life. So that is very, very important, because self-life consideration is so important that only the efficacy of the powdered form medicine of Ayurveda be important. May it be of single ingredient, as you told, or of multiple ingredient. Especially when you talked about the manjistha or even shunthi. So many things are always there, especially the dust is there. Because the person, those who are selling the this sort of crude dust, they are not in position to detect all these things, remove things. As you know that simple raw material purchasing and pulses which are coming, which we purchase from mall or a good grocery shops, they are quality controlled, because they pay attention for taking out the finest form of the pulses and all that. So goes the case with our herbal things. So thank you so much that you made things very clear that before packing, it is a must that quality control should be there, as well as the this powder form is there. Because after in a powder form, nobody knows that what is there inside.
Right, sir.
Yes, sir. So thank you so much.
Along with the Ayurvedic powders, sir, two categories of powders are being sold more. Prickly heat powder and lal dant powder. Both are being sold under Ayurvedic license because I was heading Imami R&D for so long years, 33 years. So there was lal dant powder and talcum powder, prickly heat powder. That is not directly Ayurvedic medicine, but every ingredient was Ayurvedic medicine, excluding the base. So that standardization was too fine, and we are using Ayurvedic powders in lesser finer stage, and the same is being sold as Kwatha Dravya, as a coarse powder. Government, fortunately, communicated the shelf life study before 10 years or 12 years. Before that, nobody was mentioning expiry date of powders, including Jandu also. Everybody was telling manufacturing date and use up to when this is okay. So excellent, Dr. Savita ji. Now I would like to request Dr. Guru Prasad Nillai. He is from BHU. Welcome, Dr. Guru Prasad. Welcome.
Hello.
Yes, Swagatam. You are
Yes.
clear.
Am I
Loud and
audible,
clear. Yes, yes, clear.
Okay, thank you very much, sir. Thank you very much, sir. First of all, my humble pranams to respected Pawan Kumar Sharma, sir, and other respected dignitaries, V.K. Agnihotri, sir. I think C.P. Sharma, sir, is there, and Dhiman Madam is there also. So thank you very much for giving me this opportunity to be here with you. Sir, I'm trying to start my video, but I'm unable to do that. But I think my
Then
PPT is
from
visible.
us, you have every permission.
Okay. Yeah, PPT, my PPT is visible, sir?
No, you have not shared yet. In the meantime, it was visible. Now it is not.
Okay, just a minute, sir.
Please, please. You are co-host now.
Now it is visible, sir?
No, not now. Not yet.
Just a minute. Okay. I think there is a technical problem. Previously it was shared, but now it is. Why it is not? Okay, just
Is there any problem, doctor? Dr. Guru Prasad? I hope his connection is not suitable. Dr. Guru Prasad ji. Please join, please join.
Hello, meanwhile, I can ask to Savita.
Yes, 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 sure.
Yeah, Dr. Savita.
Yes, ma'am.
Yeah, I have one doubt. Oh, you can say that it is my query. That when we study at the time Chuna and in our Samhitas, it is mentioned that when there is a season change or after monsoon, the efficacy of the Chuna will be reduced.
Ha, ma'am.
Yeah, then what can we do for that, or what can we say if that is prepared prior to monsoon and after monsoon? What should be done to that Chuna?
Ma'am, for that purpose only, they have told seasonal wise the collection of the root, leaf, stem bark, everything
Yeah, but
seasonal wise.
yes, yes. Collection is a different thing, but when tuna is prepared, generally nowadays companies are preparing as per their convenience. If they have prepared the tuna and it is in the market, right?
Yes, ma'am.
But after
Yes,
monsoon,
yes.
they are not withdrawing that product from market. Then efficacy will be altered or not? What will be the
Efficacy will
quality?
get done, madam.
Yeah,
Yeah,
what
yeah,
I
yeah.
would like to
It
answer
is-
this question, madam. I would like to answer this question. As airtight containers are there, if we put powder in polythene or extra, that is a separate issue. Otherwise, gamma radiation is also being done at the time of packaging, and the efficacy does not alter in season change because it can continue for 1 year minimum. According to the shelf life, Ayurvedic Chun's shelf life is 1 to 2 years as per government. And that has been tested that all alkaloids, glucosides, glycosides are as it is. If, but condition is there, airtight packing should be there.
Sir, but shelf life they have given the 2 years, but they didn't told that compulsory they should keep like this Tetra Pak, everything. But everyone is giving 2.5 2 years. They will keep, for example, I will give it in a, for example, in shop, I will give in a March. How they are keeping, how they are storing, that all we are not giving us, sir, commercially, what I am taking to this one, because as I am a pharmacy in charge, each shop will not tell that you keep like this, you should make like this. So here, I think it will get a little bit variation, sir.
No, as per drug control's instruction, we have to put, please store in cool and dry places. It is the
Only that much, sir.
suggestion
Only that much,
advisory
sir.
written on
Yeah.
every packing.
Sir, place is cool, place is clean, place is dry.
Dry.
But not the air. There is moisture in the air. I think that that can
Airtight
change.
container should be used. As per
Yeah,
the
that can
drug
change.
control, airtight packing should be there. Because we were selling too many powders from Jhandu, and there
Yes.
were no problem because sealing is there, polythene continuous cap sealing is there, so everything was there. And whenever leak is there, definitely that will affect the quality. That will degrade the quality.
Yes,
Yeah,
even
and
weather change, place change, everything depends, sir. That.
yeah, sometimes patient patient has taken to home, and after that there was rain, and after that, after some days, if patient will
Yes,
consume,
it
then
can
we
get
will
lost.
not get
Yes,
we
it
will
can be
not get
degraded.
result in that patient. That is the thing.
Right.
Yeah.
Dr. Suni, Dr. Savitaji, you cleared very confidently that still if slight leakage in the ceiling lid is concerned, especially Sitopaladi, Jatifaladi, Shatpushpadi, they get moistured and then उसमें गुत्थी ऐसा बन जाता है.
Especially powders containing salt.
Salt, yeah.
Lavan
Salt.
No, sir.
Ashtanga lavan.
Sugar is there.
Sugar is
Mishri.
Mishri से moisture होता है.
Hygroscopic.
Mishri, sir.
Yeah, especially sometimes there used to be some sort of small creamy also.
Silica gel bags.
Yes.
Silica gel bags are being used.
Okay. Dr. Nishan Shukla is also there.
So thank you. I would like to request now Dr. Guru Prasad Nillai for quality control of Ayurvedic powders. Welcome, doctor.
Yes, sir.
Your screen
Yes,
is
sir.
visible.
Yeah.
You can convert to slides.
Okay. Yes, sir. Yeah. So the title has been allotted to the same title has been allotted to two speakers, so it is not difficult to go with my presentation. But with a vast experience of the more than 14 years, Dr. Savita Bhosle madam has explained all the things in detail. So I will try to run with my PPTs. Okay, there is no any new concept or new things in my presentation, but I will try to convey the important things in front of this audience. Okay, so the title is quality control of Ayurvedic powders. Okay, basically what is quality? If we define the quality, then quality is the sum of all factors which contributes directly or indirectly to the safety, effectiveness, and acceptability of the product. Okay, and what is Ayurvedic powders? Yes, it is the Churna Kalpana of the Ayurveda. So we are dealing here with the quality control of Ayurvedic powders. Okay. Though the Ayurvedic powders or Churna Kalpana of the Ayurveda is very famous, and it covers the major portion of the Ayurvedic formulations mentioned in the different classical texts of the Ayurveda. In fact, in current scenario, I can say it is the least preferred Ayurvedic dosage form on the prescription of the Ayurvedic physicians and even by the consumers or the patients. Why? We see the causes. Okay. Okay. Slide run हो रही है sir? Hello. Hello.
No change
Hello.
in his slide.
Your
Your introductory slide is there. First slide.
First slide. It's now second slide. Yeah. Sir.
No, not no,
No,
not
no,
no.
no.
Okay. Okay. Now, sir.
Yeah.
Yes.
Okay. Thank you, sir. Yeah. So, according to our Ayurveda, what is Churna Kalpana? Churna Kalpana, we can get the different definitions by different our ancient scholars, and here we can see the definition by Ashtang Sangra is the Shushka Pishtaha Sukshma Tantava Patachuta Churnaha. Okay, so we can say that these ancient scholars have clearly mentioned the quality parameters through this definition. Okay, the powders, Ayurvedic powders or churna should be a completely dried product, shushka. Okay, and next one, it should be filtered through the sukshma tantava patachuta churna. Sukshma tantava pata, that means we can compare it with the mesh size of more than 85 to 120. That means very fine powder mesh size. Okay, so these are the basic quality parameters of the ancient time. Similarly, if we see that Chakrapani has mentioned that the Churna Kalpana is the Upakalpana of the Kalka, which is Kalka Kalpana, which is the one of the most important Kalpana of the Panchavidha Kashaya Kalpana, and it is mentioned that Churna Kalka eva antarbhavaniyam. Churna Kalpana, it is included in the Kalka Kalpana. Dvivido hi Kalkaha sadravo adravascha iti. Yes, the Kalka is of two types. It is sadrava and adrava, and adrava Kalka is nothing but the Churna Kalka. Okay. Sharandar has mentioned the definition of the Churna Kalpana very beautifully. And now we can see the same quality parameters as mentioned by the previous our ancient scholars. Okay, the definition is Atyanta Shushkam. Yes, completely dried powder. Atyanta Shushkam. Okay, the first requirement is the Atyanta Shushkam, and second one is the Yada Dravyam Supishtam Vastragalitam. That was should be Sukshma Tantava Patachuta. Okay, so these two parameters are here, which have been mentioned by both these ancient scholars, and we can understand by these two ancient parameters that these two ancient parameters covers most of the our quality control parameters of the this modern time. Okay, how we will discuss in the next slides. So here we all know that there are 5 categories of the churna, that is starting from the coarse powder to the very fine powder, and we know all these categories of the churna. But the new thing is that we know the term yavakuta. Okay, and in the classics, the Yavakuta means what? It is not mentioned anywhere. Okay, so this definition of the Kwata Churna, or the standard definition of the Yavakuta, is mentioned in Ayurvedic Pharmacopoeia of the India, Ayurvedic Pharmacopoeia of the Ayurveda, and it is mentioned that that Yavakuta Churna, that all particles shall pass through the sieve number 22 and not more than 10% through the sieve number 44. The product will be in the form of coarse powder, from which extemporaneous preparations of kwatas by decoctions by patients themselves can be recommended. So Kwata Churna of Yavakuta Churna of Ayurvedic Pharmacopoeia of India, and the coarse powder or moderately coarse powder, these parameters are little bit different here. So these are the guidelines available for the quality control parameters of the quality control methods of the our powders or herbal materials. And starting from the PLIM protocol that is published in 2007 by the then director Dr. Lohar, and these are the protocol for testing of ASU medicines. Previously, we were followed these guidelines for our research purposes. Then the most important quality control methods for herbal materials guidelines by WHO, which was revised guidelines of the 1998, and it is published now in 2011. And these are the important guidelines for the quality control purpose. Then ASU pharmacopoeias, yes, the Ayurvedic Pharmacopoeia of India guidelines, we have the API guidelines for the quality control methods. And again, I would like to mention that in 2080s, yes, I can mention the name of Professor K.S. Dhiman sir. I'm not, if I'm not wrong, with the efforts of K.S. Dhiman sir, these guidelines have been published by the CCRES, General Guidelines for Drug Development of Ayurvedic Formulations. There is a guideline series, and one of these series, this publication is there, available by CCRS. Okay, and then again, European Agency of Evaluation of Medicinal Products and United States Pharmacopoeia and British Pharmacopoeia and many more other guidelines are available, but we are bonded to API guidelines, CCRS guidelines, and WHO guidelines. So recently we are following these three guidelines for the quality control methods. Okay, so as we are talking about the Ayurvedic churnas, okay, mostly I am talking about the Ayurvedic polyherbal formulations, okay, not about the single churna, single drug, single herb churna. So we should know that the API part 2 from volume 1 to 4, okay, there are these volumes for the formulations only. Monographs of formulations are there in this API part 2. So here I have mentioned the summary of churna formulation in API part 2. See, the total number of formulations in all these 4 volumes are 202, and out of these 202, only 31 monographs are related with the churna formulation. So this number is very less, very less. Okay, but the Ministry of Ayush Ministry and with the help of the PCMH continuously trying to upgrade these monographs, and they are working hardly for the next volumes on the formulations of the churnas. So these guidelines are available from the CCRS, and there are 21 test parameters. Okay, total 21 parameters for the quality control of the churna and Kwata churna. Okay, and this is nothing but Kudinir churnam for the Siddha. So we will go one by one. Already, Dr. Bhosle ma'am has explained all the details about the these all characteristics parameter, but I will go in a very brief. Okay, so these characteristic parameters are for the confirmation or to confirm the identity, quality, and purity, and these are mostly classified into 4 major parameters. These are pharmacognostic parameters, then physicochemical parameters, then microbiological and chromatographic parameters. So first one is the pharmacognostical study, which includes the determination of foreign matter, then organoleptic parameters, then fluorescence analysis and microscopic study of powder. Okay, determination of foreign matters. Yes, our churna preparations or Ayurvedic powders should be free from foreign matters. Okay, like the other parts of the same plant or the other plant parts, or we can say the sand particles or stones or the molds or other insects with their excretas and other we can say the physical the poisonous or hazardous chemical exudates. It should be free from all these foreign matters. Okay, so organoleptic parameters, yes, already madam has explained with some wonderful examples, so I'm not going in these parameters to explain, but I can say that
please continue your flow as detailed, sir, because
okay, sir.
New visitor, new audience will be benefited by that. So please go ahead.
Right. Okay.
Don't think that it is repeating. Yeah. Please go ahead.
Okay. Thank you, sir. So in organoleptic parameters, we can say that our Acharya has mentioned that Rasagandha Varna Gandha Varna Rasodbhavaha. Okay, it is not only applicable for the Avleha. It is not applicable only for the any specific dosage form. It is applicable for the all the dosage forms of the Ayurveda. Okay, and that is nothing but the Gandha Varna Rasodbhava. That is nothing but the organoleptic parameters of that ancient time. Okay, so that these Gandha Varna Rasodbhava, these quality organoleptic parameters, what does indicate? It indicates that our herbal drugs are rich of phytochemicals, rich of having phytoconstituents, and they are fresh. They are freshly collected, and so that they are there with these fulfilling these parameters. So color, order, taste, appearance, and textures, these are the organoleptic parameters for the which included in the pharmacognostical study. Next one is the fluorescence analysis and microscopic study of powder. We will discuss in the next slides. Sorry. So fluorescence analysis. So what is fluorescence analysis? Why it is important? See, some chemical substances exhibits fluorescence phenomenon. Okay, so in this case, in our case, the plant phytoconstituents have the same properties. Okay, with the help of UV light or even in the visible or daylight, the most of the natural products exhibits fluorescence phenomenon. Okay, that means they indicate that there is a presence of specific chemical compounds which are performing the fluorescence phenomenon. Okay, which are responding to the light and shows the fluorescence phenomenon. Okay, but sometimes with adding the other reactive reagents like concentrated HCl or K2 Cr2O7 or sodium hydroxide or silver nitrate or many bromine water or iodine, methanol, these reagents may activate these natural products or these phytoconstituents into their we can say their derivatives which have the properties of the fluorescence analysis. So this is the one of the most important quality parameter, or so we can say the quality parameter of the pharmacognostic study. So here I have mentioned the two examples, that is the fluorescence analysis of the Amritadi Churn, and here you can say you can see the different colors of the in visible daylight and UV light of the powder drug with different reagents. And same with the Samasharkara Churna example of the Samasharkara Churna is there. So next one is a microscopic study. Yes, microscopic study is important. Why? It is to study the specific microscopic characters, to identify the adulterants, and to confirm the presence of ingredients of a polyherbal powder. Okay, so with the help of microscopic study, we can even confirm the presence of a specific ingredient of a polyherbal powder. For example, if your sample has a cork cell or stone cell or scalariform xylem vessels and trichomes, that means your sample contains guduchi. Okay. You can see in the image that the first image is of cork cell, second one is a stone cell, third one is a scalariform, xylem vessels, and last one is a trichomes. Okay, so guduchi has these specific microscopic characteristics. Okay. So next one is after pharmacognostical study or parameters. Now we are coming to physicochemical investigations, and these are the standard pharmacopoeial. There are some standard pharmacopoeial methods which include the total ash, acid insoluble ash, alcohol and water soluble extractives, loss on drying, pH, etc. So total ash, for what we are doing, total ash indicates the presence of, or indicates the presence of inorganic content in your drug. Acid insoluble ash, to measure the presence of, or measure the amount of silica in your product. Okay, alcohol and water soluble extractives. These for the measurement of the, or these are the for the we can say the consistency and the amount of the specific chemical compounds present in your herbal drug. Okay, next one is loss on drying. Yes, it reveals the moisture content of your product, and yes, pH is also there. So these are some physicochemical investigation. Next one are physical characteristics of powder. Yes, these physical characteristics like bulk density, tap density, angle of repose, Hausner's ratio, and compressibility index or Carr's index. These all show the powder flow. Okay, powder flowability. So why it is important? Because the flowability of the powder is important for the mixing, for the packaging, for the purpose of transportation of the manufacturing process. Okay, so the bulk density, tap density, both defines the flowability of the powder. And angle of repose, it is considered that if it is angle of repose is under the 30, then the flowability is good. If it is in between the your 30 to 45, then there is a cohesiveness, and if it is more than 55, then it has very less flowability. So for that flowability purpose, angle of repose is important. Similarly, Hausner's ratio, it should be less than 1.25, and the smaller the compressibility index or Carr's index, the better is the flowability of the powder. So for this purpose, bulk density, tap density, angle of repose, Hausner's ratio, compressibility index are the important parameters for the powder. The next one is qualitative phytochemical investigations. Yes, there are different tests for alkaloids, tannins, steroids, glycosides, flavonoids, saponins, carbohydrates, terpenoids, proteins, using different reagents and tests. Okay, for the qualitative phytochemical investigation. I'm not going in detail about all these details of the procedures of these tests. So next one is the determination of toxic components. Yes, it includes the heavy metal determination. Next one is a microbial limit test, then pesticide residue study, and aflatoxin determination. We will see one by one. Okay, why heavy metal determination is important? Okay, nowadays many publications are raising their heads. Okay, by defaming the Ayurvedic herbal products. I'm not talking about the herbo-mineral products. We all know that herbo-mineral products have the major portion of heavy metals, as we have deliberately mixed the heavy metals there, but in a specific chemical compound or after the specific procedures of the Ras Shastra. But in case of herbal medicines, if we see that there are many examples that heavy metal contamination in herbal medicines and the related toxicity. Okay, so why here I have quoted one example, and this is the very latest recent publication published in Frontiers in Pharmacology, which is very reputed journal. The title of the publication is Heavy Metal Contamination in Herbal Medicines, Determination, Comprehensive Risk Assessments and Solution. Okay. So what are the sources of heavy metal contamination? Exactly how the contamination of the heavy metals happens. Okay. So there are different sources of the heavy metal contamination, and these sources are the anthropogenic sources and natural sources. Okay. So anthropogenic sources includes the cement manufacturing, then smelting plants, then waste incineration, fossil fuel power plant, then inorganic fertilizers, pesticides, sewage sludge, and your fly ash, then wastewater treatment plants, then chemical industries, traffic and other emissions, then ore extractions and other industrial processes. And natural sources are the volcanic eruptions, then wind blown dust particles, then the sea spray, then aerosols and natural rocks. Okay. These sources are the responsible for the heavy metal contamination in herbal products, herbs. So by the way of air, water and soil, all these heavy metal ions uh, uh, contaminate the our raw material of the herbs. Here you can see the figure, all the details of the uptake, mobility and translocation of ions in heavy metal ions in plant parts have been depicted in this figure beautifully. So you can go in details about this article, which is published in Journal of Toxics, MDPI publication. And the title is Medicinal Plant Growth in Heavy Metal Contaminated Soils, Responses to Metal Stress and Induced Risk to Human Health. So in the API, we can see the list of heavy metals, and these are lead, mercury, arsenic, cadmium, chromium, zinc, copper, nickel. And there are some permissible limits for the heavy metal contents. And for the lead, it is 10 ppm, arsenic 3 ppm, cadmium 0.3 ppm, and mercury 1 ppm. So we should be conscious about the heavy metal determination with the help of AAS, atomic absorption spectroscopy technique, or even we can use the ICP-AES, that is inductively coupled plasma atomic emission spectroscopy. These two techniques are there for the determination of heavy metals in our samples. Next one is the microbial limit test. Yes, microbial limit test. There are also permissible limits for some microbes and some yeast and molds, and these have been clearly mentioned in our API. Next one is a pesticide residue study by GC-MS system, and here mostly we deal with the three types of pesticides and three classes of pesticides, and these are organochlorine pesticides, organophosphorus pesticides, and pyrethroid pesticides. And in API, the limit, permissible limit or acceptance limit have been mentioned in detail. Here you can see that there are number of tests for different pesticides, and the limit not more than limit of detection should be less than 10 ppb, okay, by GC-MS system. The next one is aflatoxin determination. Yes, aflatoxins are mucotoxin, which are produced by the certain types of fungi, and these are very toxic substance. And with the acute exposure, we can see some mild symptoms like headache, nausea, or other some mild symptoms. But with the chronic exposure to aflatoxins, it can lead to even the liver cancer, okay, or growth failure in children, or immune suppression. Therefore, the aflatoxin, the permissible limit for the 4 aflatoxins have been mentioned in our API, and these aflatoxins are B1, G1, B2, and G2, and there are the mentioned permissible limit. Yes, the chromatographic analysis. These are the quality control techniques or fingerprint techniques. for the qualitative and quantitative estimation or evaluation of our herbal products. And this includes the TLC, then HPTLC analysis and HPLC, etc. Okay, there are many chromatographic analysis, sophisticated techniques, we can say, for the analysis, chromatographic analysis of our herbal products. Okay, by using these chromatographic analysis, we can evaluate, you can estimate the quantitative and qualitative estimation of some important biomarkers from our herbal drugs, or we can say it is the at the simultaneously we can identify the a wide range of our markers or phytochemicals in our given product with the help of these chromatographic techniques. It is a separation technique, and it is the most required technique for the quality control of our polyherbal formulations. So some other sophisticated techniques are there, like TGA, thermogrammetric analysis, then FTIR is there, spectroscopic techniques are there, including FTIR, Raman, infrared, near infrared technique, then NMR is there, then mass spectroscopy is there, then liquid chromatography along with high resolution with your MS, and gas chromatography with the tandem MS. Okay, these sophisticated techniques are also there for the quality control of on the basis of WHO GMP parameters. So here I have mentioned some few examples, good examples of the publications. You can go with these publications here. Almost all the quality control parameters have been followed by the authors, and they have very well explained each and every step of the quality control methods in their publications. And this is the pharmacognostic, then physicochemical and chromatographic characterization of Samasharkara churna. It is published in JIM journal. So next one is the estimation of eugenol from Ayurvedic formulations by using RP, reverse phase HPLC. Yes. So here we can see that the reverse phase HPLC, this is the validated chromatographic or chromatographic technique method. Okay, validated. It is validated. It is very difficult process. I think first we have to validate or develop the technique as per or following the your ICH, International Conference on Harmonization, ICH your guidelines, and it includes the precision, accuracy, repeatability, and reproducibility. Okay, if you are validating some methods to for any specific test, any to estimate or to check the any specific biomarkers in your formulation, herbal formulation. So this is the most beautiful example of that sophisticated analytical technique, that is RPHCLC. It is very costly. It is time-taking procedure. Therefore, the HPTLC is always preferred along with densitometric method. Densitometric HPTLC method is preferred over HPLC because of very cost-effective, very short time required there, and cost-effective method is there. Then accuracy is there, precise. It is very precise, and reproducibility is there. Therefore, your HPTLC is preferred over the RPHCLC. But this is the best example that eugenol, like your essential oil containing your phenolic compound, that is eugenol, and estimation of eugenol by using the HPLC in different churnas like Chaturjat churna, then your Jati Phaladi churna, then Sitopaladi churna. How beautifully they have quantified this eugenol and standardized or quality. They have produced the quality control parameters for these churnas. So this is also the good example of publication. So next one is the method validation and quantification of major biomarkers in Mahasudarshan churna and Ayurvedic polyherbal formulation through high performance HPTLC chromatography. In this article, they have quantified, estimated the major biomarkers in Mahasudarshan Churna, and they have targeted the your mangiferin, then your ursolic acid, then your quercetin, then your curcumin, and I think one more biomarker is there by using the your HPTLC method. Okay. So next one, nowadays Kwatha Churna is very famous churna, your type of our churna, because of the AYUSH Kwatha Churna. The Minister of AYUSH has provided the AYUSH Kwatha Churna during the pandemic, COVID pandemic, and due to which the AYUSH Kwatha Churna is now very famous in market. At the same time, the Ayurvedic Formulary of India has published the formulary specification of AYUSH Kwatha Churna, and in the 2020, and recently in 2022, one publication is there in IJTK, International Journal of Traditional Knowledge, which is the publication of the CSIR. In this publication, authors from the CCRES have published the development of quality standards of Triphala Kwatha Churna with its ingredients through HPTLC and mass spectroscopy. Okay, so this is the also another example of the good publication. So regarding stability study of the Churna, these are the test descriptions for the accelerated stability study, that is for 6-month stability study, and here you can see the test parameters and number of samples. That total 18 parameters are there for the accelerated stability study, and required number of samples for that, because some of the parameters requires test in a triplicate manner. So description at the starting, then after 3 months, and after 6 months, then foreign matter for the 3 times, powder microscopy for the 3 times, then particle size by sieve method for the 3 times, loss on drying for the 3 times, then total ash for the 3 times. Similarly, we can see quantification of upload oxygen by HPTLC only at the starting point, then your heavy metal analysis only for the starting time. So these are the test parameters for the stability study of the Churna, that is accelerated stability study. And finally, here, this is the most recent example that why quality control is important. Yes, stringent quality control can save the future. Of course, I'm talking about the Ayurvedic formulations, but the example is very right that recently it is published that the Medan pharmaceutical the India says that samples taken from the Medan Pharma from linked to Gambia dates meet required standards. Okay, and similarly, the news in Times of India was the Gambia linked drugs are found to be of standard quality. Government takes on who? Okay, so these cough syrup has fulfilled the quality parameters of the I think Indian Pharmacopoeia or WHO on WHO quality parameters, and therefore this news is there. So we should maintain the quality of our Ayurvedic pharmaceuticals following all these quality parameters which have been published by our authorities, and with these words, I'm very much thankful to Pawan Kumar Sharma sir and all the other members on this conference, international conference series. Thank you very much.
Yeah. Excellent, doctor. Excellent. You have covered almost all the things. Excellent. And that quality control is very necessary for this modern era, as we are playing with the quality. Generally, SSI, small scale industry, is not maintaining any quality control parameters. So our message should be stricter than our words, that everybody should maintain the quality control. Agnihotri sir. Now our next webinar is there after Agnihotri ji's comments. So I am changing the YouTube link. Agnihotri sir.
Thank you, Dr. Guru Prasad ji. You have very deeply and very simple way touched all the areas regarding the quality control of the Ayurvedic preparation in form of churn, especially all sort of analysis which are supposed to be carried out is a must. Then only we can have a better quality product in a form of churn. As far as their this shelf life is